There is an animal alive today that may have been born before the American Civil War. It weighs more than 80,000 kilograms. It has trillions of cells, lives for more than 200 years, and yet does not appear to suffer the enormous cancer burden that basic probability would predict. It is the bowhead whale. And scientists recently discovered something unexpected inside its cells. The whale does not seem to survive by simply destroying damaged cells faster. It repairs broken DNA unusually well. Meanwhile, another animal has developed a completely different version of the same strategy. The naked mole rat is roughly the size of a mouse, yet it can live for decades. Researchers found that small evolutionary changes in one of its proteins make a DNA repair pathway behave differently. When scientists transferred parts of this biology into other animals, they saw signs of better health and longer life. Together, these discoveries are pushing longevity research toward a provocative question. What if one reason we age is not simply that our DNA gets damaged, but that our ability to repair that damage is not good enough? Your DNA is being damaged constantly. Ultraviolet radiation can alter it. Environmental chemicals can damage it. Normal metabolism generates reactive molecules that can attack it. And every time a cell copies its DNA before dividing, mistakes can occur. Most of these problems are repaired. If they were not, life would be impossible. Cells contain multiple repair systems specialized for different types of damage. But one form of damage is particularly dangerous: a double-strand break. Imagine DNA as a long instruction manual made from two connected strands. Now imagine both strands being cut at the same position. The cell has to reconnect them correctly. Get it wrong and genetic information can disappear, move to the wrong location, or become rearranged. Enough mistakes can contribute to cancer or loss of normal cellular function. This creates an enormous problem for long-lived animals. The longer an animal lives, the more opportunities its cells have to accumulate damage. A larger animal also has more cells. So you might expect giant, long-lived mammals to develop cancer constantly. They do not. This contradiction is known as Peto's paradox. The bowhead whale may be one of nature's most extreme examples. Its maximum lifespan exceeds 200 years, making it the longest-lived known mammal, while its enormous body contains vastly more cells than a human body. Researchers compared bowhead whale cells with cells from humans and several other mammals. At first, they expected the whale to have extremely powerful mechanisms for preventing damaged cells from becoming cancerous. Instead, they found something surprising. Bowhead whale fibroblasts actually required fewer cancer-promoting genetic changes to undergo malignant transformation than human fibroblasts. That sounds like exactly the opposite of what a 200-year-old animal should have evolved. Then the scientists looked at DNA repair, and the mystery began to make sense. Bowhead whale cells repaired DNA double-strand breaks more efficiently and with greater accuracy than cells from several other mammals. They also showed fewer mutations after repair. The researchers then searched for what was helping them do it. One protein stood out: CIRBP. Its full name is cold-inducible RNA-binding protein. Humans have CIRBP too, but bowhead whale cells contain unusually high amounts of it. CIRBP appears to gather around broken DNA and help organize the repair process. It can promote the binding of repair proteins to DNA and protect broken DNA ends from being degraded before they are reconnected. Think of a broken chromosome as a snapped cable. Before technicians can reconnect it, the exposed ends need to remain intact. If pieces are lost before the repair crew arrives, the cable may still be reconnected, but the information it carries could be corrupted. Higher levels of CIRBP appear to help protect those ends while the repair machinery works. When researchers added bowhead whale CIRBP to human cells, DNA repair improved. Both major double-strand break repair pathways became more effective. Large insertion and deletion errors decreased. Chromosomal abnormalities also declined. When researchers reduced CIRBP inside bowhead whale cells, the opposite happened. Repair efficiency fell and errors increased. Then the researchers tested something even more interesting. Could this protein influence longevity outside whales? They increased either human or bowhead whale CIRBP in fruit flies. Both versions extended adult lifespan. The modified flies were also much more resistant to ionizing radiation, which causes severe DNA damage. That does not mean CIRBP will extend human lifespan. A fruit fly is not a person, but the experiment matters because it moves the story beyond correlation. High CIRBP was not simply something scientists happened to observe in a long-lived whale. Changing CIRBP altered DNA repair and survival in experimental systems. And the whale is not the only long-lived animal pointing toward DNA repair. Consider the naked mole rat. A normal laboratory mouse lives only a few years. A naked mole rat can live for several decades despite being similar in size. It also shows unusual resistance to many age-related diseases. In 2025, researchers reported that naked mole rats possess an altered version of cGAS, a protein better known for helping the immune system detect DNA in places where it should not be. Normally, cGAS is part of an alarm system. If DNA appears in the cytoplasm outside the nucleus, it can signal infection or cellular damage. But cGAS also interacts with DNA repair. In ordinary mice and humans, part of that interaction can interfere with repair. The naked mole rat version behaves differently. Several amino acid substitutions changed the protein in a way that improved its ability to support genome maintenance. Researchers found that this naked mole rat mechanism enhanced DNA repair and delayed aspects of aging. The really important pattern is that bowhead whales and naked mole rats did not evolve exactly the same trick. One has high CIRBP. The other has unusual cGAS biology. Both arrived at a similar solution: protect the genome better. Evolution appears to have repeatedly favored improved DNA maintenance in species that live extraordinarily long lives. And that agrees with an even larger observation. Across mammals, the rate at which somatic mutations accumulate tends to scale inversely with lifespan. Long-lived species generally accumulate mutations more slowly. The body seems to pay for longevity partly by investing more heavily in genome stability. But this raises a dangerous temptation. If better DNA repair is associated with long life, why not simply turn every DNA repair pathway up as high as possible? Because repair is not automatically harmless. Cells make trade-offs. Some damage should cause a cell to stop dividing. Some heavily damaged cells should undergo programmed death. Repairing a cell that is already dangerous could theoretically allow it to survive when elimination would have been safer. DNA repair pathways also interact with cancer biology. A tumor cell with extremely powerful repair systems may become resistant to chemotherapy or radiation. Enforcing repair proteins to remain highly active could disrupt other cellular processes. More repair is not necessarily better. The goal would be better repair at the right place, at the right time, with the right level of accuracy. That is why animals such as the bowhead whale are so valuable. Scientists are not just looking for proteins that are abundant. They are trying to understand how evolution tuned entire systems without creating catastrophic side effects. There is another fascinating clue in CIRBP's name: cold-inducible. In human fibroblasts, the researchers found that lowering culture temperature from 37 degrees Celsius to 33 increased CIRBP levels and improved one form of DNA repair. Bowhead whales spend their lives in extremely cold, Arctic and subarctic waters. It is possible that an ancient response to cold eventually became part of a genome protection strategy. But this does not mean cold showers or ice baths will reproduce bowhead whale longevity. The experimental temperatures were applied directly to cultured cells. Human body temperature is tightly regulated, and chronic hypothermia is dangerous. The finding is mechanistic, not a lifestyle recommendation. Scientists are now exploring several ways DNA repair could eventually be targeted therapeutically. One approach would be increasing protective repair proteins. Another could involve stabilizing proteins that become less effective with age. Researchers could potentially enhance the recruitment of repair machinery to DNA breaks or improve the accuracy of specific repair pathways. But the challenge is enormous. DNA damage is not one thing. Ultraviolet damage requires different machinery from oxidative damage. Single-strand breaks differ from double-strand breaks. Damage in dividing cells creates different problems from damage in neurons that may survive for an entire lifetime. A drug that benefits one tissue could harm another. And aging itself is not caused only by DNA damage. Mitochondria change. Proteins lose quality control. Stem cells decline. Epigenetic regulation shifts. Senescent cells accumulate. Inflammatory signals change. Organs communicate differently. DNA repair is one piece of a much larger system. Even so, it is an unusually fundamental piece. Every cell depends on the information stored in its genome. If that information becomes increasingly corrupted, every downstream system can eventually be affected. This may also help explain why DNA damage connects aging and cancer so strongly. Aging requires cells to function for decades. Cancer requires cells to acquire combinations of changes that allow uncontrolled growth. Better genome maintenance could potentially reduce both problems. That is exactly what makes the bowhead whale so fascinating. It is not merely long-lived. It is enormous. A gigantic body living for 2 centuries should create an extraordinary number of opportunities for cancer. Instead, evolution appears to have found ways to preserve genomic stability across that enormous number of cells. There are hints that exceptionally long-lived humans may also carry advantages in DNA maintenance. Studies of people reaching 105 years and beyond have identified genetic patterns associated with DNA repair and genome stability. But extreme human longevity is complex. No single DNA repair gene explains why someone becomes a centenarian. Immune function, metabolism, cardiovascular health, lifestyle, environmental exposure, and genetics all interact. There is no CIRBP supplement proven to slow human aging. There is no cGAS treatment approved to extend lifespan. And nobody should attempt to manipulate these pathways based on animal experiments. The field is still trying to answer the most basic translational question. Can evolution's longevity solutions be borrowed safely? Nature has already performed experiments lasting millions of years. The bowhead whale evolved one solution. The naked mole rat evolved another. Humans evolved our own compromises. Scientists are now reading those evolutionary experiments like engineering manuals. Not because they expect humans to live 200 years by copying one whale protein, but because these animals reveal which biological systems are capable of being improved. The most important message is not that scientists have found the longevity gene. They have not. It is that extremely long life does not necessarily require cells that never become damaged. Damage is unavoidable. The difference may be what happens afterward. A short-lived animal repairs the break one way. A bowhead whale repairs it more accurately. A naked mole rat changes how repair proteins behave. And over decades or centuries, millions of tiny differences in repair quality could add up. Aging may be shaped not only by how much damage life throws at us. It may also be shaped by how well our cells keep fixing the instructions.